This has been the first session and has been very basic research. It's going to be changing as we progress and become more work on mice and then humans and then in the clinic. But so this first session is very basic research centered. So perhaps there are technical questions, because I know that in the audience there are scientists and PhD students, and maybe there are questions about deep science, and perhaps after that we can talk about more general things. I can see that there is one question here by José Antonio Enriquez.
Hi, I want to make a comment first about your one statement that you made.
Myself?
Yeah. Because you got, you bring one of the sentences that I always teach, that nothing is, makes sense in biology if you don't consider evolution. But you make another statement that I disagree. Because you say that nothing makes sense in pathology if you don't consider aging. And I think that there are gerontologists here, but if there were an audience of pediatricians, I will tell you that this is completely wrong. And all the infection medicine will tell you that. So this statement may apply to a particular group of diseases, but not to diseases as a whole.
I see the point. Chronic diseases. Chronic diseases. Well, I don't know if anyone in this symposium is going to talk about what is called the programming of aging, about how the intrauterine life affects aging, how our childhood affects aging.
But this is another point. I mean, defining aging, we are not really common agree in defining aging. So all the intrauterine life, so aging doesn't start when you make the silent. So aging starts when you finally, at least from the medical point of view, when you finally finish the development, not only in the intrauterine, but also in the acquiring sexual maturity. I think that there is a tremendous debate about this point, but and that's the second point that I think that is important for the audience, is that aging is not a disease.
Nobody said that.
No, but you can whip all the diseases, and as many of the treatments that you show in many models, all the animals almost die very close to the same age. So maximum lifespan expansion is really very, very minimal. What is true that is the health lifespan is really improved quite a lot. So this is, I think that this kind of, there are points that might be interesting to discuss for this kind of audience.
Felipe, please.
Just a note on that. Yes, I think we all agree that the aging process is affected by things that happen all through even intrauterine life. There's no question about that. The effects of life course is all affecting aging. When we're talking about aging, is the downstream effects of that. We're not eliminating that there is an effect before. Yes, there is, but we're focusing on the facts or the things that happen as you reach the later part of your life.
Comments from you about this, Linda? Another question here, please. Okay.
Good morning. It's a question for Dr. Partridge and for Dr. Cuervo. This is in regards with a drug that has a bimodal mode of action, which is metformin. Have you ever worked with metformin, trying to influence the RAS pathway and the mTOR pathway? And for Professor Cuervo, have you explored the role of mitochondria, especially the abnormal mitochondria, which are, they have to be removed, and maybe this is one great problem because it produces some antibodies and so on. Thank you.
Metformin's a really interesting drug, and the expert on its effects on aging is sitting in the front row here near Barcelona. We've done a little bit with it in flies, and I think this may be a case case where flies don't quite compute when it comes to mammals. So metformin extends lifespan in worms, but it seems to do so mainly by affecting the metabolism of the gut bacteria. So if you feed the worms on dead bacteria, they no longer show an increase in lifespan in response to metformin. We've tried it in flies, and it activates AMPK, which is one of its actions, but it's quite a broad spectrum drug. It does several different things. And in flies, it doesn't extend their lifespan at any dose that we can find. And the mouse work, really, you should be asking CNIO. We haven't done anything there. So I think it's a very interesting drug. It clearly has multiple different modes of action, and it's not clear which ones are critical for its geroprotective effects as opposed to its ability to prevent type 2 diabetes.
So on the same line, because we've been working with Nick and Derek Huffman, looking at the effects of metformin in mammalian cells, and we can see, as other groups have shown, that it activates macroautophagy. As Linda mentioned, the question is out there whether the effect in the mice depends fully on autophagy or not, and we're talking about doing autophagy deficient animals and try to do those kind of interventions. But as Linda mentioned, you will hear all about metformin from Mir Barzilai later. And regarding the questions about the mitochondria, I think that's a terrific question. I focus for today in this particular form of autophagy that degrades single proteins. You have the other form of autophagy, this macroautophagy, that when it degrades mitochondria as a whole, it's called mitophagy, and basically that one also decreases with age. So I fully agree that the inability to turn over your mitochondria at the normal rate leaves them compromised, and you have more damaged mitochondria and higher production. But the interesting thing that I think we are learning is that you don't have to eliminate your whole mitochondria. So you do that as a stream. When everything is too damaged, you go for this mitophagy. There are other ways in which the mitochondria can either extrude their bad proteins, and Heidi and María have shown beautifully this vesiculation happening. And even for this pathway that I talked today, as you know, most of the mitochondria are encoded in the nucleus. So those proteins cycle through the cytosol before reaching the mitochondria, and then they can go straight again if they are damaged. And we found that this chaperone-mediated autophagy contributes to their degradation. So in a way, you preserve your mitochondria, but you are changing the enzymatic load and maintaining it healthy by degrading them when they are still in the cytosol. So I think mitochondria is a great organ, and another example of how proteostasis interact with other parts like metabolism in these pillars of aging.
Thank you, Anna. There is a question here, please.
I'm Juan Lahuerta. I'm a clinician interested in neurogeriatrics. First of all, I want to thank the speakers panel because you have given to us a truly intellectual feast this morning. And my question is addressed to Dr. Serrano, again, relates to the senescent cells and the response they produce on fibrotic response as well as a chronic inflammatory response. My question was, and although I think has been partially answered by Dr. Cuervo, what is the origin of this abundance of senescent cells? Is it because we produce more, or is it because our immune system is not able to remove them? I think that's why Dr. Cuervo, when she showed that graph indicating that autophagy and T-cell response seems to be correlated, and perhaps both are taking place at the same time. But I would like to have your thoughts about this.
It's a great question, and unfortunately, this is an open question. We still don't know what is the reason why when we are old, we accumulate more cells. Most of the people in the field suspect that it's the immune system, that it's the failure of the immune system to remove these cells. But this has not been demonstrated in a rigorous manner. It's a suspicion more than evidence. I hope very soon we will know the answer to that. I mean, a lot of people is working at trying to answer that question specifically about the immune system. It's a good question. We have another question here. Actually, two questions. This is.
I wanted to start by echoing what was said a moment ago. I think we've had fantastic presentations to start the discussion. Particularly, I wanted to ask Anna Maria, in your diagram, you gave a really lovely description of how autophagy plays so many important roles. You said that autophagy, if you have impaired, reduced autophagy, it can lead to a compromise of energy production. But I wanted to ask you a little bit about directionality, because I've always thought of it as impairment to energy production, compromise of mitochondrial function, perhaps leading to a reduction in because autophagy is an energy-hungry process, leading to an impairment of autophagy. So I guess it's true in all of these processes that there is complex bidirectionality, and this probably applies to several of the other presentations we've heard. But I wonder whether you'd like to comment on that, please.
Yeah, no, that's a terrific point. I mean, for sake of the time, I probably oversimplified, but the idea is that even autophagy is an energetic hungry process, as you mentioned. Not all the forms of autophagy are so demanding. So if you compare the requirements for macroautophagy versus endosomal microautophagy and chaperone-mediated autophagy, those ones require less energy because you don't need the formation of a new organelle. So when you measure that, and that's probably where you activate chaperone-mediated autophagy late in starvation, because by then your energy is very low, and this is a process that can help. So in a way, you still have a positive balance considering what you are mobilizing. The interesting thing, I mean, as I said, I fully agree that it's going to be directional. At some point, if your ATP levels are too low, these processes are going to be compromised, and the acidification of the lysosomes might eventually get compromised. But the interesting thing is that because autophagy can mobilize very valuable energetic source, you can do lipophagy, you can degrade lipids, and that's going to be a great source of energy. So of course you are using some energy to do that, but you still have a positive balance. Same thing with glycophagy that some labs are studying. You mobilize a lot of glycogen in a single compartment in a very short time. So I think probably the cell is smart enough that figure out that you invest a little in this cleaning crew, but they are really going to give you a lot of energy. But you are completely right. At some point, these things are bidirectional, and everything might get also affected. Yeah.
Thank you. Please go ahead.
Thank you. Well, first of all, I have to agree. I was about to get a Stendhal syndrome with the talks in the morning. It was just amazing. Secondly, I tend to agree with you, Manuel. I do believe, and we can have a debate after all, that nothing in pathology makes sense except in the rate of aging. Essentially because every early onset disease is an opportunity for pleiotropy, for antagonistic pleiotropy to kick in and actually generate resistance to that disease. But anyway, my question is that one pattern that seems to emerge out of all your talks is that there are several different mechanisms that have been acting over and over again across very different species. And this is, I will read your paper, Darío, later on, but, which I haven't so far. But the question is directed to you. You have all these skill fishes and you detect all sorts of, I suppose, genes and pathways that are linked to aging in them. And my question is, have you checked whether these same pathways and genes have been linked to aging in mammals, fish, primates, flies, and so on and so forth? Up to which point all of this is shared?
So the functional adaptation, thank you for your question. The functional adaptation that we use for the killifish comes from shared aging pathways. So when we can call the go pathway as aging, this is because of functional annotation done on experimental systems. So it's based on conservation. Excuse me? Sort of?
A lot of conservation.
A lot of conservation. Well, I mean, at the gene level, and we can really tell apart variants that have been, hitting aging pathways that have been positive selected from the ones that are under neutral evolution. They have a very distinct genomic signature when you look at population. So, yeah.
I want to take the opportunity to ask you, Darío, you have talked a lot about genes and how the genome is amplified, but what is the impact of that on the transcriptome? Have you compared the RNA levels of different genes between, how do they compare?
This is a wonderful question. So indeed, we have a look at where this amplification occurs. And one striking factor that we observe is that the more a gene is expressed at a given time in life, the fewer are the nested transposable elements that are in its introns specifically. Because for annotation reasons, we are better at detecting intronic transposable elements rather than transposable elements closer to the regulatory sequences. But if a gene is highly expressed, it actually will have small introns. And if a gene has low expression or is expressed later on in life, in non-developmental processes, then actually even its introns are expanding due to transposition, active transposition.
Very interesting. Let's, in the back, and then we can hear. Please go ahead.
Yes, thank you. First of all, I want to thank the panel. Great morning, great talks. Just a question for Dr. Cuervo. You mentioned, or maybe for the rest of the panel, because it's partially related with another question that they posed before. You mentioned about the genetic manipulation and autophagy, but also you mentioned at least diet. Can you tell us a bit more? Is caloric restriction or fasting your experience? Just caloric and fasting and autophagy?
Yeah, people are going to hate me because this is right before lunch. But yeah, you are right. So something that has been known for many years is that calorie restriction is one of the interventions that extend lifespan but also health span. That is what we want at the end. And from the point of view of autophagy, there are very elegant work showing that autophagy in part is contributing to this beneficial effect of calorie restriction. So basically you have, during calorie restriction, when you reduce the amount of calories, as John was pointing out, you activate this process of autophagy. So in the case of macroautophagy, the one with the vesicles trapping mitochondria, you get a much better regulation. For example, in the liver, insulin blocks autophagy, glucagon activated, so you get your very nice peaks of autophagy with the time. In the case of chaperone-mediated autophagy, we are about to submit the work showing that what you increase is the threshold. So your autophagy, your chaperone-mediated autophagy during calorie restriction, basal is higher already during the whole time. We all realize that calorie restriction is something that's very difficult to implement. And then the laboratory of Rajat Singh, also at Einstein, have shown that the same beneficial effect, at least at the level of autophagy, that you get by reducing your calories to 60%, that is not going to be easy, you get it maintaining the same calories, but separating the time of feeding. And I think in a way that kind of makes sense. You activate autophagy when you don't have food. So you activate it when you sleep, and we sleep each time less and less, so not much time for activation of autophagy there. And also we are continuously snacking or picoteando for the Spanish part. So the problem is that we don't allow our autophagy that should be activated in all your organs at least once a day to clean up what is around. We don't really allow that because it's so linked to nutrients that we kind of have it compressed. So I know that endocrinologists might not fully agree with this idea, but at least experimentally in the animal models that Rajat was using, the animals are much fit. Not only they have better regulation of insulin, but they also have, the frailty is dramatically reduced. They have less incidence of cancer, and now there are many other labs doing this food clocking, this separation, and it seems that part of the beneficial effect is because of activation of autophagy.
Thank you. Neil, please.
First, thank you for this dream team, and I think it's clear to the audience that you said really the same thing, that biology, that aging biology and this biology can be targeted, and that's why we're so optimistic. I want to ask specifically Darío, because the transposome and the line animals were very important for evolution, but work from Guptov and Sedevi lab showed that actually there's an activation with aging of some of the line 1 that actually can drive senescent autophagy. So I'm asking you the cause-effect relationship. Maybe they live shorter because they are activating your models.
Yeah, thank you very much. So I want to make a distinction. So what we see is germline expansion of transposable elements. Our elements that are vastly expanded are L2s. I don't rule out that what you are pointing out may be happening in killifish. Indeed, we know from transcriptomics data that there is an activation of certain classes of transposable elements, REX1s in particular, which are not the number ones that are amplified at the germline level. But we're going to be looking more into this. So of course, this is work in progress. We have unveiled this kind of like connection between genome size expansion and demography, if you wish. So weaker selection. So our model currently is that there is less of a genome guarding in a way that unleashes. So this lower genome guarding is due to this drift event, and this unleashes this expansion of germline expansion of transposable elements. Now, what you're asking is specifically to do with what happens during ontogenesis rather than phylogenesis, and this is definitely something we will be looking into.
Are there killifish in the zoo of Madrid?
I don't know.
It would be interesting to check the telomeres
Yeah,
in the
it would be
different
interesting.
populations.
Yeah.
There is a question waiting there in the back. Pablo, please.
I have two questions. The first one comes from the talk of the Partridge, but it's more a general comment because my impression is that right now we have many approaches to reduce aging in a more like molecule-wise. We have senolytics, we have telomerase inducers, we have metformin, we have nicotinamide riboside, but then the Partridge came with an important point, which is the combination of all these drugs, because it seems that everybody is using them single, but I think it's very interesting, very important that we start combining, because that way the idea is that we can reduce the side effects and also improve altogether. So I think, have you, in the case of Linda, you just measured some of these combinations, but I guess there should be some research going on with combination of other pathways that are not the only ones that you mentioned. You mentioned metformin also. Have you checked, for example, autophagy inducers or senolytics in your combination strategy?
So I agree with you that combinations are very important. And the way I think about it is a bit like the outcome of, there's been a recent clinical trial of a polypill against cardiovascular disease conducted in northwestern, I think, Iran. And they found that they would give just in the general population, so primary prevention. And there were 4 drugs in this polypill, and they really got a massive reduction in the incidence of cardiovascular disease as a result of using it. Now, it's not clear from that study which constituents of the pill, it could be a single one that was actually really important, but I think it's an interesting example of a clinical trial with a major mixture of drugs, just as primary prevention. So that's where I see this sort of thing going. And I think the experimental work will come. But obviously if you're doing, lifespan and health span study in mice, and you've got your single controls, your double controls, and you rapidly end up with a very, very major enterprise indeed. So I think it's going to be selective. It's going to be a question of picking actors that look as though they would combine in a beneficial fashion because they're targeting different aspects of the aging process. But I think we're going to see a lot more of that. And probably it's somewhere where the invertebrates are going to be very useful to start with, because you can make progress much more quickly and more cheaply, and then look at whether they affect the same aspects of the aging process in mice.
This is a very interesting change of paradigm, because traditional pharmacology thinks that when you combine drugs, you multiply the toxicities rather than reducing the toxicities. So this is very interesting. We have one question here, please.
Okay, so this is for Linda. I'm still trying to wrap my head around the implications of your study of the persistent benefits of rapamycin. And I think this is like fundamental, huge step for the aging field, because it suggests that we have to rethink the biology of aging, and that we can, it's sort of not a linear process where you basically reset the at least gut to a younger level and it stayed young. And I think for Felipe, for everybody, the implications of that work, and it's, you know, maybe not specific to TORC1 inhibition, but that you can reset the clock and it stays, and whether it's all just autophagy or there's some epigenetic component, or I'm not sure yet, but just curious because I think that's a huge finding.
Yeah, and especially as Anna Maria said, since so many of the interventions that seem to ameliorate one or more effects of aging do activate autophagy. And since at least in Drosophila it seems clear that once autophagy is up, it stays up. If you induce it directly, not using rapamycin or any other method, it could actually have quite widespread significance for any intervention where the major actor is an increase in autophagy.
Now, can I say, actually, I agree. This is kind of a paradigm shift about several things that have been said today, this morning, actually, that something has been percolating into the research recently, which is actually precisely what I think was Manuel who said, when you take different drugs, the concern is always that they're going to amplify the negative things. What we have seen in the aging field is that these hallmarks or pillars that have been described actually interact with each other in a positive way. So when we affect, no matter which one, you affect the other ones in a positive way, which is something completely unexpected in my mind, and it's really, in my view, the most exciting thing happening right now. And indeed, that means that we can take different drugs that affect different ones of these pillars, reduce their levels, the level of the drug, right? So as to have less side effects, and will improve the whole basal line, as you said. I think it's really earth-shattering.
But also, it's not just reducing the level, you take it away completely and you just stay.
You just set the
That's
time. Yeah,
really,
absolutely.
interesting.
Absolutely.
The memory.
There have already been hints of that, sorry to interrupt you, with rapamycin. So, particularly Dr. Manuel did an intermittent treatment with mice and found that he didn't have the positive control of the chronic treatment, but it was effective. It improved the health of the mice. So that there have been hints for a while that it might have this persistent effect, and it clearly does.
Right, but we never had the biology to understand what it was, right?
Well, one of the hallmarks or pillars of aging is epigenetics, and perhaps this memory to the drugs is connected with epigenetics to produce a permanent change. Please go ahead.
I have a question for the entire panel. Do you consider that aging in mice or flies that live in the wild and have a constant lifetime that does not change is the same than aging in people, in human beings that cheats their own nature and have extended their lifespan? Do you think that the biology of aging for human beings who have successfully cheated nature is the same than in other species that have a fixed lifetime that has not changed in millions of years?
If I may, there are many answers to that excellent question, actually. There's a lot of things in there. So first of all, in terms of expanding lifespan in humans, what we have expanded is the mean lifespan, but not the maximum. So we also have a fixed lifespan, which we have not been able to change much. We might be able to, but we have not. Also in terms of whether different species have a fixed lifespan, yes, they do. But the variability within the species, within a cohort, even for example, C. elegans living in the same dish, the first one to die, that timing is shorter than the distance between the first one that dies and the last one that dies. The heterogeneity at which they die is enormous, even in species with the same genetics. So whether or not this is applicable to humans, I think that there's not as much a difference in there as you might think. And one thing that's kind of related, but I always want to point out, is that we have all heard the sentence of, yes, 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 we have cured cancer so many times in mice, but not in humans. So let me just tell you one thing that we have as an advantage in aging research, which is when you try to cure cancer in a mouse, you're usually inducing cancer in a mouse. And and that's a less than optimal situation. You induce cardiovascular disease in a mouse that normally does not have cardiovascular disease, Alzheimer's, the same thing. Those models have very strong limitations. When we're talking about aging, we're talking about animals that are allowed to age normally. We're not manipulating that aspect. We're not manipulating the biology that we're trying to modify. We're just trying to modify their normal biology. And that's why I'm optimistic about being able to pass from the mice to the humans.
If I may add one point to this. Actually, I don't think that humans have defeated nature more than domesticated animals have. So, if anything, actually, domesticated organisms have done it even more so than humans have. So, when we use model organisms, we work with laboratory organisms that have been bred, that have been, a mouse in nature doesn't live as much as a mouse in the lab. A fly in nature doesn't live as much as a fly in a test tube. So I think that in that respect, we're doing a good job in using protected organisms, just like we are protecting our own environment.
Yeah, I think you did not get my message or my question. I work in mice, so I'm not affected by working in fish or mice or flies. It's just that I do think that, and as you said, there is an heterogeneity, so the median in human is not that important. That human beings live longer than a century ago, and with that comes a slew of diseases that did not exist before. Whereas killifish or elephants probably live the same time now than in the 19th century. They do go through aging, but the aging process in human now include diseases that were not necessarily known, maybe not in the 19th century, but a century, 1,000 years ago. And in that sense, molecularly, the two processes of aging, the one that is natural aging, and the one of humans and domesticated but not non-domesticated animal species is different. And I think it's important to consider the medical implication of what we do in aging, because I'm working only in mice.
Yeah. In relation to this, I wanted to ask to Maria about what she thinks about the clocks. Now, a very important thing is the aging clocks, and one of them is the telomeres. And more than aging clocks, morbidity or predictors of morbidity, because now there are many, and some of them are very accurate, and telomeres is one of them. What is your opinion about this?
About clocks?
About the clocks in general, about the power that they have, about how they can impact on the clinical trials, because we can, theoretically, we can measure how biologically old is a person and see if the therapies have an impact on that, because...
I think that's a very interesting point. I mean, how you can use also biomarkers of aging to measure the toxicity of maybe drugs that are being used to treat cancer, or I think that is not regularly done, and I think it's one of the interests of understanding aging also, to see which could be the effect on aging, on the molecular pillars or hallmarks of aging, of treatments that are being commonly given to people for treatment of cancer or other diseases. I think it's very, very interesting.
Yeah.
That would go with the interest of studying aging, to have better biomarkers, and telomeres may be one of them. Methylation of the genome. I don't know. I'm sure there is a panel of biomarkers that I think that's something. Autophagy, of course, I was looking to Ana Maria, but I think maybe that's something interesting also to convey to medical doctors and to industry, that this could be interesting to see the impact of drugs that are being used for treatment of other diseases on aging.
Well, actually, there's one example that I've heard many times is that, for example, you can only donate kidneys after a certain age. However, if you look at the functional kidney and the molecular aspects of the kidneys at people at different ages, well, you see some kidneys of 35-year-olds that are completely shot, and some kidneys from 80-year-olds that are very, very healthy. Mm-hmm. So we throw away a lot of potential donors of kidneys, which are absolutely necessary just because of their chronological age. which is the only parameter we can use. So if we could have clocks like this that could be tested in a fast manner so that we can do it within the time that it allows for transplantation, that already by itself would be a fantastic improvement in health.
Okay, let's have 3 more questions. Please, the microphone here, because Dr. Jose Viña has been waiting for a long time.
Thank you, Manuel. It's been a great pleasure being here with you. I will follow for once the rules and try, as Felipe Sierra said, to think of general ideas rather than picture. And therefore, Felipe, I have a question for you. So in one of your first slides, you showed that the damage accumulation of Alzheimer's comes very low and then goes up. And you say, I understood that you said there's no damage accumulation in aging, and therefore damage protection in anti-aging. And my question is, if you, instead of seeing a order 1 kinetics, i.e. linear, you see order 2 kinetics, i.e. the damage causes more damage, then you'd have something that approaches that you could adjust it quite to the line you showed, the 2 lines, which can be one exponential line. So do you really think, having said that, do you really think that we have damage accumulation, or thinking like Manuel said of the fetal programming of aging, all that kind of thing, or not? Or aging is still, after what I've said, in your opinion, not associated strictly with damage?
Yeah, I have to agree with you that that second order kinetics could be important too. Especially in Alzheimer's, they have been shown not as much for A beta but for tau, that seeding a little bit of tau leads to more tau accumulation. So that is certainly an alternative explanation if you wish. However, I would argue that because this happens with so many diseases in so many different instances, like the example I gave with cancer, cancers are so different, caused by different genes, in different tissues, and so on. And they all come up exactly at the same time. The chances that it's just because of second order kinetics is certainly there, but it's not, I don't think it's the only explanation.
Thank you.
Thank you. Any burning questions? Here, okay.
I'd like to ask a couple of questions about rapamycin. The first for Linda. Have you read Mikhail Blagosklony's theory that in humans females have higher maximum lifespan than males because of a lesser basal mTOR activation? And have you confirmed this theory with mice, that maybe males have a better response to rapamycin than females? And also for María, what is the reason that autophagy correlates with a better immune function? And do you think that's the reason why rapamycin can be used to improve vaccine response?
Rapamycin in mice, you probably noticed from the graph that actually at a given dose, the extension in the food, a given amount, the lifespan extension is less in males. So they show a dose-dependent increase in lifespan, but it's of much lower amplitude than females show at the same range of dietary concentrations of the drug. And the reason seems to be that males detoxify it more actively. So if you actually look at the serum concentration of the drug, it's lower in males for a given dose in the food. So answering your question is complicated. I think we don't know at the moment if for a given effective inhibition of TORC1 in the cells, we're getting a lesser or greater response for autophagy in males than females. I don't think that question's been answered because of this complication of them responding differently to the same dose.
So regarding the role of autophagy in this improved immune response, this is not work from my lab, it's from Fernando Smathian lab, but basically what they have shown is that the two main types of autophagy, macroautophagy and chaperone-mediated autophagy, seems to be contributing in a different way to the immune response. In the case of macroautophagy, and this will be related to rapamycin and what they saw in centenarians, what it's doing is really shifting the energetics of the T cells during the moment of activation. As you know, T cells are look more like cancer cells in a way, so they are very dependent on this glycolytic process, but at some point, in order to get the activation. So autophagy, by excluding mitochondria, so you don't degrade your mitochondria anymore, so you have more energetic, and not you go for lipids. Apparently, you can get the boost in energy that you need for that activation. So he thinks that in this case of centenarians, they were also responding better because of better energetics. And in that case, I fully agree that seeing the effect that rapamycin is having in many processes, including autophagy, although it could also act on autophagy as an energetic mechanism and the metabolic pathway per se, that's probably why it might have a beneficial effect in the immune response. For the other pathway, for chaperone-mediated autophagy, Ruth Baldor has shown that you need that form of autophagy in T cells to eliminate inhibitors of activation. So when you need to activate a T cell, you normally have them in a break. And there are these proteins that H and ERCAN that are maintaining it in a break. At the moment that you need to activate, you have to remove the inhibition. And they show very nicely that chaperone-mediated autophagy does that. As you get old, your chaperone-mediated autophagy is not able to do that, so you have a higher inhibition of your activation of T cells. This was very T cell centric because that's where their lab worked, but there is beautiful work done also in dendritic cells and presentation and the relation with autophagy. So I think the fact that autophagy is such a universal function for most of the cells might impact in many components of the immune response and came out with that beneficial effect.
Thank you, Anna. We are going to stop here. We are going to have, I'm sorry for those of you that still have questions, because there are still 5 more sessions and opportunities to debate and to make comments. So we should stop here, and I want to thank all the speakers and the audience for the questions. Everybody, good job, everybody. I forgot to mention that we should be back, we should convene at 2:30. Instead of 2:15, 2:30. Right? Yes.